All right, so this is actually a pleasure to introduce Professor Graham Ball. He is a good friend, good collaborator, and eminent scientist working in the area of bioinformatics and machine learning. And especially, he has good grasp and understanding of omics data set, which is a very rare combination, which we see people who can appreciate the statistics and bioinformatics and machine learning approaches. So I'm sure all the participants are really going to get very enriched experience from this session. Professor Graham has conducted a couple of big data workshops, which has been highly successful. And I'm sure today's session is going to also enlighten our participants about how to take some of the big data set and perform machine learning AI kind of approaches to narrow down the leads. I'd like to emphasize the way we are discussing the entire proteomics workflow from today morning. You have done the experiments to identify the targets, did some validation, but still you are ending up with set of proteins which is all looking good to you, but you can't narrow them down, like, which is the best panel which could predict that this is, good biomarker for a given disease. For instance, while comparing breast cancer samples, Dr. Shrikant might have found, 25 proteins. Now, how to narrow it down to make the panel of 3 or 5 proteins, which is most influential to be developed in the test. And that's where all the machine learning tools comes into the play to do various iterations and see that, what is the weight of each of the predicted biomarkers for that test. And Professor Graham has really been helpful to some of our project to enlighten the best notes possible, which can be major hub for looking at that disease. So Professor Graham, welcome to this session. Thanks for accepting our invitation. And next 1 hour is all yours to give participants an overview of this field.
So thank you.
One small request, if you could make your presentation a slideshow so that we get a complete screen.
I've set it as a slideshow.
On top of the screen, you can see end slide, and yeah, there's a presentation mode that you could select, the 4th option.
Okay. Sorry, just bear with me. Sorry, I'm just trying to.
No problem.
Sure. Apologies.
No problem. So you take your time. It just happens. Just have to click on share and then the presentation, make it full screen mode.
I see. So if I click share now.
Yeah, I can see the presentation. Yes. Do you see the 4th option, which has a small icon which says the presentation mode?
That's on the bottom right? Bottom right.
Bottom right side, where you have an option to zoom.
Do you mean this? No.
No, actually, what I presume that whether you have double screen, because sometime in the Mac, if you have double screen,
yes,
it shows like that.
Okay.
So is that? Can you see that now?
Yeah, we can just make it a bit full screen, sir. There is a 4th option called yeah, it's coming up now.
Actually, alternatively, Professor Graham, if you have double screen in Mac, you can stop the other screen to be seen. Then this whole thing will be coming on the full screen.
I see. I need 2 screens for what I'm doing now. So, so can you see my presentation now?
We can, but it is not in the full screen on the.
Okay. And now.
Sir, could you see there is an option for zooming your presentation? Just before you zoom, at the right side below, there is an icon that you could see like a floating panel.
Yeah.
No, don't zoom it. Just before the zoom option, there's a floating. We just can make it 4th one. Yeah, that's the one that you're pointing now. Yes, correct.
So can you see my slideshow now?
Yeah, no. Same issue.
So I think that's the issue. So what I have to do is, what I'll do is I'll keep it on. If I change the view, I can set it as reading view.
Now it is
Yeah,
good.
Sanjay, this is fine. This is fine, I think.
Yeah, it's
So
fine
that's
now.
all. I think.
Instead of
Yeah.
the double screen issues. Yeah. Good to go, Professor Graham.
Okay, thank you. So once again, thank you very much for the opportunity to present here. It's my great pleasure to be visiting India again, and unfortunately it's virtual, not in person, but that's the nature of the world we live in at the moment. But it's a great pleasure to present and work with Professor Srivastava, and you know, he's a good friend, he's a good colleague, and a very eminent researcher who it's my honor to work with. So I am a bioinformatician, and what I need a lot of is data. But we have to consider data in certain ways. So what I'm going to talk to you today about is about an overview of the subject, is about some of the issues that we face with omics data and big data generally. And then I'll talk a little bit about some of our solutions, and then I'll go on to do a demonstration of a piece of machine learning software called Orange. So if you search in Google for Orange data mining, then you will find this software, but I'll show you the webpage. So this is kind of an overview of the subject of bioinformatics, and I've tried to cover the full spectrum. So from something that's purely mathematics right the way through to the omics technologies. So the first part of this is biostatistics. And we should always work on good statistical principles when we are doing any analysis of any data. So we can use parametric and non-parametric statistics. We need to test hypotheses. So for example, we might want to look at the ability or the difference in a particular ion or molecule between two different groups. We might want to test the hypothesis that a particular molecule is associated with a particular state in a population or disease. Getting a little bit more advanced, we might want to mathematically model the relationship between those molecules and an outcome. And that's pretty much what we do with our machine learning approaches. We're developing and making models of a system. We might want to map or structure the data. We might want to do an ordination. So when we map the data, we want to maybe see outliers, we maybe want to see subgroups in the data, maybe structural elements in the population. And that's an important to provide understanding about what's happening in the data. We might want to model temporal events. So for example, survival analysis might tell us what the risk is for a group of patients. And again, that's a modeling approach, and we can and have used mass spectrometry data to model events in cancer, to identify risk factors. And we might want to look at trends through a population to see if there's a group of individuals who have predictive elements that behave in one way, or a group of individuals that have a different set of predictive elements that make them behave in a different way, and to see what proportion of the population that covers. So a next aspect is starting to move towards the biological sciences, and that's systems biology. So here, what we're saying is, can we use omics data and methodologies to start to understand a little bit more about the biological system? We are all a very complex soup of molecules. We have around 22,000 genes. These code for 200,000 proteins and downstream metabolites and a whole set of processes. So theoretically, every protein could interact with every other protein. So it's unraveling this complex jigsaw that helps us understand about the biology of disease. We're not just a list of markers, it's how those markers interact with one another. So systems biology is a way of achieving this. systems biology as a way of understanding the interrelationship between things. And it may be the interrelationship between tissues, it may be the interrelationship between molecules. But ultimately, there are computational methods we can use to study this. There is one approach which we use called network inference. Network inference identifies how things interact. And we can explore pathways through this network inference approach. So, for example, if we have a model of a pathway, we can see molecules A and B linked together. If we wanted to look at protein-protein interactions, we could use something like string, which would give us both the known interactions as well as the hypothetical binding interactions. So it's all about understanding this pathway and this kind of complex tangle of interactions and biological processes. So yeah, things such as string, gene mania, ingenuity pathway analysis, KEGG and Metacore will help us using databases to understand how things might interact from a list of features. So since you've said you may have 25 interesting features, you might want to understand what their interplay is, how they interact with one another, what processes they're involved in. And we can use machine learning as well to do this. So we've developed a network inference approach which allows us to model pathways and understand what the drivers of a pathway are in a biological system. Another aspect is computational biology. So this is where we're using computer methods to understand and identify biological features. So we use these methods to look at ways of finding the best set of features that answer a particular question. And we can do that with modeling. So what is the best correlate between a protein and a clinical question? What is the best combination of correlates or combination of proteins that predicts a clinical question? And we can start to answer some of these questions with machine learning. So machine learning is a form of artificial intelligence. It's really you could also argue it's a very complex, multi-dimensional, non-linear regression analysis. But the difference between regression and machine learning is that machine learning feeds back iteratively. So it learns from its mistakes. Errors are fed back to optimize the model. Examples of machine learning include artificial neural networks, which are the method I tend to use, but also things like random forests, support vector machines. And there's a phrase which has come out lately called deep learning. And I'll talk a little bit about that later, but it may not always be suitable for high-dimensional biological data. One of the other areas of growth, which is quite important, is integration of multi-omic studies. So incorporation of transcriptomic, proteomic, metabolomic, and genetic information. And this is going to grow more and more because more and more data is being generated for biological systems. We also might want to identify biomarkers. So, for example, features that can be physiological, molecular, genetic, that indicate a particular group or outcome or answer to a question. So, for example, we've got areas of work in the diagnosis of latent tuberculosis. We've got areas of work in the diagnosis in breast cancer, in brain tumors, in lung cancer, where we want to understand what's going to happen to a patient or whether they have a particular disease or not. The main area we're looking at is bioinformatics, and this is effectively how data is managed. So bioinformatics is really the use of informatic methods for biology. So it's in the interrogation of databases, it's the management of data, alignment of data, normalization of data. And really sort of covers databases, location, temporal data, population data. So for example, some of the methods that have been described, things like Protein Pilot, MaxQuant, things like Swiss prot are all bioinformatic methods. It's basically getting information from a database and processing data to achieve a particular result. One example of this is sequence. So, for example, we need to align sequence data to the human genome to understand which genes or which part of the human genome a sequence will cover. And we can use these sort of bioinformatic workflows in things like R, Python, Anaconda, Bioconductor, and often they're deployed on Unix or Linux systems. So bioinformatics is a way of interrogating data, helps merge data, process data, and gain information about the ontology and function of a particular target as well. We can also use molecular docking, which is called chemoinformatics, and that might be, for example, to discover a drug which blocks a certain molecule or a certain biological process. The last element of this is bioinformatics. Sorry, is omics. So this is where the data is generated. And you can see how this really links to bioinformatics, because we need to, when we generate data, we need to understand it, we need to store it, we need to process it, we need to align it. And this is getting more and more complicated. So, for example, when we consider single cell sequencing now, you can have 100,000 cells, each of which have been sequenced with 200,000 pieces of information. There's also developments around localization and spatial proteomics and flow cytometric-based proteomics, where we end up with very large amounts of information which we have to disentangle. So really, the omics is the fundamental pillar and bedrock behind a lot of the other things we do. We need to understand the strengths and weakness of the data. We need to understand how we deal with the complexity of that data, and actually getting something useful biologically out of it. So we know we need a whole range of different methods to bring all of that together. And we can use things like chemoinformatics, systems chemistry, as well, to understand drug targeting. So what is artificial intelligence? Well, artificial intelligence is the theory and development of computer systems which are able to perform tasks normally requiring human intelligence. So the human mind is very good at spotting patterns in things. It's very good at inferring knowledge and extrapolating knowledge from abstract subjects. Machine learning seeks to try and replicate that, but we don't really have true AI. We don't have the sort of AI that we see in science fiction. We have effectively a synthetic AI. So examples are Alexa. Sorry, my Alexa is about to kick off now when I've said that. But Alexa is a system which understands questions and can do act in response. But it's not true artificial intelligence. You cannot ask it to do something that which is much more in depth. So what we have is effectively simulated intelligence, and many people who claim they're using AI aren't really using AI. So an extension of AI is machine learning, and this is perhaps more realistic. So this is the study of computer algorithms that improve automatically through experience using learning. It's a branch of AI, but effectively it's ultimately an extension of mathematical modeling. So what we have is a training data set which is used to make predictions or decisions without being programmed to do so explicitly. So it's learning a solution. We might have a complex proteomic signature, and from that signature we want to identify the best set of features that predict cancer, for example. Now we can use a supervised approach where we tell it for a set of cases which have cancer and which don't, but we're not sort of trying to program it from first principles. We're getting it to learn a solution. And so effectively, we're using this to identify structures and patterns using these computational learning techniques. So AI is used in a number of ways in biology. So it can be used in ordination, the mapping of structures in data. And these are an extension of clustering methods and principal components and methods. So for example, random forests are an extension of a clustering method. We can use Kahonen networks to structure populations. We can interrogate data. So we are using machine learning. We're using an artificial neural network to interrogate data to find the best set of features that predict something we want to know. We can mine data. So maybe we have 10,000 variables or 100,000 variables. We want to find which is the best set of those. We want to simplify that complex data down into a few key features. We want to model. So we want to represent information, and we want to be able to disseminate that. So we want to be able to share a model that other people can test. And in reality, what we have is combinations of all of those. So we have combinations of every method to try and achieve what we want to achieve within a given data set. So these methods are applied to big data, and big data has challenges. Now, big data in biology is considered to be hundreds to millions of variables. So we can have an individual might be profiled using, let's say, an exon array which has a million megabases on it. An individual might be measured with a multi-omic technique where they have potentially millions of variables. And often the number of variables will exceed the number of cases that we have. So we have many more variables than we do cases. And it's just not feasible to get millions of cases and do sequencing or mass spectrometry on them easily or inexpensively. Actually, big data in biology is quite small compared to other cases. So some of the engineering big data sets have 100 million or even a billion features in them, whereas biological data might be 100 to 500,000. So what we are using big data for in biology is because it gives us a deep overview of the biological system. We can simultaneously measure the expression of every gene. We can simultaneously measure individual transcripts or exons in the whole genome. And if we want to systematically evaluate the genome, that means we can consider every possible feature and make sure that the things we're finding are the most robust. So we don't miss anything. And that's the problem with smaller studies. We might miss something that's important just because we haven't included it. So yeah, we want a system where a large number or there's a high proportionality of coverage where every gene, transcript, or protein is represented. And we get a comprehensive overview of the system. Now, there are various challenges. We have a challenge about the quality and replication of data. We can't have degradation in a sample. We have to have ideally equal reproducibility for every component in the data set. And this is where some of the early expression arrays fell down and were problematic. They were very good at measuring certain things, but very bad at measuring others. So if you were to analyze them, all you end up doing is finding a set of things that are good measurements. You don't find the core biology of the system. So we have to have an equal reproducibility. Everything has to be of good quality, really. If we put rubbish into our model or into our analysis, we will get rubbish out. We will get things that are just random noise. So ideally, we need a good representative data set as well. We don't have to have enough so that every individual we have the same number of individuals as we do variables, but we do have to have a reasonably good representation. And from our powering, around 40 individuals per class is what we need. So this is also a challenge because it's highly dimensional data. So it's a challenge because in high dimensional data systems, where we have a large number of variables, it's sometimes difficult to find the key features. So what happens is the dimensionality of the data can mask what's really important. If you look, it's a bit like looking at the night sky. You can see 100 million stars, but you can't see the planets orbiting around one single star. And which one do you pick to look at? So it's very, if we look at a very complex space, it's hard to identify the key feature easily. And so what's happening is the dimensionality is masking the importance of a given marker or feature. We refer to it as looking for a straw-colored needle in a haystack, but it's perhaps more like looking for a straw-colored needle in a field full of haystacks. We also have challenges around computational time. So if we were to develop a classifier from a data set which had 60,000 dimensions, and we were to run a 10-gene panel on that with 50 random sample cross-validations, so 50 splits of the randomized splits of the data, then we would need 3 times 10 to the 47 different models to consider all possible combinations. That would take quite a lot of computational time. Now, we're getting over some of these issues now by working on GPUs, graphical processing units, effectively creating a PC-based supercomputer where we can run a lot of analysis in parallel. Another point is we need to have generalized models. So we want a model or a classifier which should serve the general population. It should be broadly applicable to the population or a broader population and not constrained by a large number of conditions. So, for example, we've done one of my former PhD students who's now working in Stanford, Gopal Dondale, worked on estrogen receptor positive breast cancer. And in the course of his studies, he found somebody who was looking at estrogen receptor positive breast cancer in patients aged 55 to 58 who are HER2 negative, grade 3 stage 2 tumor with overexpression of Ki67 BRCA and VEGF. This is the problem. Many times scientists will want to control everything to such an extent that it doesn't give a realistic solution. And the problem with this sort of study is that we would find in the UK maybe 4 cases like that per year. So it solves the issue for them, but it doesn't actually solve the issue for the wider population. Also, we see sometimes that the number of parameters in a model or in a system exceeds the number of cases. So, for example, we've done a study from the Berry data on tuberculosis, and Berry had a panel of 293 transcripts that would predict tuberculosis absence or presence, but for only 120 cases. So the problem there becomes, well, in that model, every individual has their own gene and could still predict 100%. So it's challenging. We have to keep the model simple as well as keeping it generalized. Another challenge in biology is non-linearity. Biology is not linear. Genes are switched on or off. They can have step functions. There can be interactions between components and complexity. So we have to use something that's not linear. If we use a linear method to analyze biological data, then anything we discover will be constrained by that linear paradigm. So why we use a neural network is because it allows us to add multiple levels of non-linearity into that solution. Another challenge is one, and this is important for big data, is the consistency of findings. So big data is very prone to false discovery. If we look at 50,000 variables and we have 5% false discovery rate, that still gives us 2,500 false features. So we have to use robust approaches to find consistent findings. So often methods will be criticized because the same method finds different answers for different data sets, or a different method finds different answers for the same data set. So this is consistency issue, says that they're not really appropriately powered and that we're not really finding the key features of the data. So it might be that an individual data set doesn't have enough statistical power as well. So these might be false discoveries. So we've developed approaches where we actually look at multiple data sets and multiple splits to try and find the most robust elements that are consistently presented across those different systems and data sets. So this is our hypothesis. So this is a well-known British suite, and let's say we have a set of individuals. The red individuals might respond to a drug and the yellow ones might not. So we can then profile those individuals. Here we're using expression array data sets. And we then use a neural network. So a neural network is effectively a matrix of interaction method, a matrix of weights, where values that feed in at the top are mathematically modified to give a value out at the bottom that allows us to predict. And it learns by updating those weights in response to error. So with this approach, we can effectively pick the key transcripts off the expression array, and we can see whether treatment A or treatment B should be applied. And we have thus a stratified population. So we're using the machine learning to identify the features. Now, this diagram overly simplifies it, but essentially to do that, what we do is look at every single feature on the array or on the mass spec profile, and we test the performance of that feature for the question we're asking. And that gives us a rank of performances. And from that, we can start to understand what the key predictors are of the system. We can also use that in a stepwise fashion where we add iteratively features into the model and see how the model performance changes. So a neural network is a simplified version of a biological neural network. So it has an input and an output, or effectively a dendrite and an axon. And the values, as I said, are fed in which transform the input values through to output values. So expression levels, protein measurement levels are fed in, and that predicts whether it's cancer, not cancer, or it predicts a continuous value. So we use this for pattern recognition and to identify a set of biomarkers or indicative biomarkers. We can use it to recognize patterns and reduce noise. We can predict based on previous data, so we can predict well for new cases. And it can learn, it can generalize, and it can cope with nonlinear and complex data. So our approach is we train the data, feeding a single input or transcript or protein into the model, testing an output, and then that's compared to what actually happens, and that gives us an error, which is then fed back into the algorithm. So it's a cyclical process where we make a prediction, test a prediction, use the error to feed back into the model to update the model, and keep cycling around until we have an acceptable model. So the error drops in this as we train, but there's a problem in that the error drops to a minimum and then increases. So what's happening here is the model is ceasing to be good at predicting for unseen cases. It's memorizing rather than generalizing. So what we want to do is stop the model at the lowest point of the red line. And to do that, we test the network on an unseen data set and then stop after the model's failed to improve. So using that approach, we were able to stratify and stratify cases and stratify populations based upon models. So the next part of this demonstration is, I'm just going to stop sharing and restart sharing. So we use one of the methods that's very easy to use and relatively straightforward as a good starting point for doing machine learning is Orange. And Orange is a machine learning data mining toolkit that can be found at orange.biolab.si, or if you Google Orange data mining, you can find it. And what's good about this is you don't have to be able to program for a start. There are also some quite good tutorials on and videos on how to use it. And it's quite, as you'll see when I start to go through it, it's quite it has quite a wide range of tools and methodologies that you can employ. So it's a good starting point to learn and do some initial analysis. I would say, work on Orange. Then the next level of knowledge is in a system called Weka. So if you search for Weka and Waikato, you can find another toolkit that also has a lot of video support, which can show you and teach you how to use it. And then I would say probably after using Weka, then if you want to take this a lot further, then I would start to use Python and things like TensorFlow, Keras, Pandas to start to develop your own code in the machine learning space. So I would just, without too much ado, I'm just going to go to Weka now. Bear with me. So where are we? Orange. So you should be able to see an Orange instance, and it should be a blank screen. Is that the case? Hello?
Yes, Graham, go ahead.
Okay, perfect. So Orange has a whole lot of visual tools. And the way we work this, it's a bit like something like Open Workbench. We just drop those tools onto a breadboard, onto a whiteboard. Each tool is an icon, which you can see here on the left-hand side. So, for example, if I want to load a file, I select the file widget. If I want help on that, I can just right-click and get help, and it tells me all sorts of ways of using that widget. So what we want to do with file is we want to set where the file is. So I can use normal Windows, or in this case Mac, to identify where that course is, file is, and load it in. What I can also do is start to look at the features and set which ones are numeric, which ones are categorical, and potentially edit them as well to change the type. So that's the starting point. We need some data to work with, and this is molecular data from a test for sepsis that I'm looking at. So you can see here we have a whole range of tools. We can import SQL. We can import Python scripts if we want to. We can impute differences. We can transpose. We can construct feature rules. So there's a whole range of things we can just do in this basic set. And we can also save the data. So the next thing I want to do here is I want to select the columns I'm going to use for my machine learning. So what I do is I drag the select columns feature in, and I then drag a line from file into select columns. So let me just check that. So that's one of the things we have to check is the link. And so what we want here is data coming in and going to data as an input to select columns. So now you can see what we have here is a way of selecting the things we put into our machine learning. Now, you might want to create multiple instances of this. So Professor Srivastava before was saying about maybe selecting 25 features. Now, you may want to create 25 of these and use each one as a separate input, so we can then cross-evaluate those. So here, what I'm going to do is I'm going to select these 5 features and predict here disease positive or negative. We've imported our data, we've fed it from there into select columns, and from select columns, we've then selected the things we want to take forward. So we can then move on to some of these other toolkits. So for example, we might want to do an unsupervised analysis. So what we can do here, for example, is a principal components analysis. So I'll take our data from our selected columns into principal components analysis. And if I click here, I can select the number of components I use. So here we have a principal components analysis, which uses, 5 components from our data ultimately. Remember we had 5 inputs, but let's select, say, 3. So 3 gives us 84.8% coverage or explained variation in our system. So that gives us the things to select. We might want to then put the results of that into a data table. So we might want to get that data and do something with it. So if we drag data table on, we take the output from the PCA, put that into a data table, and you can see now where we have our individual eigenvalues for the principal components, and we can see we have the measured values as well, so we can look for correlations there. So we can also do things like we might want to do a scatter plot. So we go to visualize and drag a scatter plot there, and we drag the results from that to scatter plot. Now we need to select in scatter plot what we're going to plot. So if we plot here, oops, it's not yet. So we need selected data to date. So we need data to data. Yeah. So these linkages are important. If they are not correct, then the system might not work. Now you see that's gone from a dotted line to a solid line, so that's now working. If we go to scatter plot, we're now plotting principal component 1 against principal component 2. And you can see that those two on their own don't really separate out the data too much. So we can then look at a linear projection, which is another way of handling these PCA results. And you can see here the line's dotted again, so I need to just check that. I'm going from data to data. That's now fixed it. And now you can see we have a three-dimensional plot, and we can see better separability here in the three-dimensional. We can select various things here in the principal component. Now we can move that around. I'm not sure if we can rotate it. That's something I need to look into a bit more. But essentially, you can see we're able to create a workflow that goes from a file right the way through to plotting and data. Right, something else I might want to do is a hierarchical clustering. So for that, I go again to unsupervised, and for hierarchical clustering, I want the distances. So I take effectively our data and calculate the distance between every point and every other point in the data. And here we can select the various methods of distance. So for example, for continuous data, we might choose Euclidean. For genomic data or sequence data, where we have a base pair status, I might want to use Manhattan. So once we have the distances, then we can put those into a hierarchical clustering. So again, I drag the values from the distances into the hierarchical clustering. And now you see we have a cluster dendrogram. And we can change the different linkage methods. So for example, we can have a single linkage, we can have an average linkage, we can use complete linkage, or we can use Ward's method. And in this case, I think Ward's method probably gives us the best set of features. So that's immediately covered two, well, they're not really machine learning methods, they're quasi-machine learning methods, but they're kind of the early start of that process. So I might then want to start to use some machine learning. And so to do that, we go to model. And here you see we have a range of different methods that we can use. We have decision tree methods, we have random forest methods, support vector machines, logistic regression, naive Bayes, neural networks. And so we have a number of methods we can use here to start to explore the ability of these markers to predict disease presence. So let's start with a logistic regression. I think that should work. Yeah. Yeah, so we're taking our selected columns out and putting them into logistic regression. And we can double click on that and select the sort of conditions we use in that logistic regression. At any point, I'll just reiterate, if I need to help on the thing I'm doing, then I can right click and come up with help, and it'll tell me how to do things, what the background is behind the method. You can see here, for example, I've got a link out to logistic regression. And so that's quite helpful for learning as well. So here I'm creating a logistic regression, and then I need to evaluate. So there's an evaluate parameter set here, and I want to test and score. So what test and score does is it allows me to test the performance of the method we're using. And to do this, I need the output from the logistic regression, but also I need data coming from the data selection as well, because I'm comparing those two methods, one to the other. So if you look now, I've got from my logistic regression, I can see that I have an area under the curve of 0.967. So that's needs to be as high as possible. And we have precision and recall methods. So these should be as high as possible as well. I can also do various things here like cross validation, repeating unseen cases, leave one out, prediction of classes, and model comparison area under the ROC curve. But I have a whole range of parameters I can select here. I don't have time to go into the parameters or the basis of these methods, but again, there's a lot of support and help that you can gain from Orange to do that. So what I have so far then is I have a principal components method, a clustering method, a logistic regression method, and I'm testing the logistic regression method in this system. So I can also add here maybe a neural network. So again, I'm feeding data in from the select columns. I'm setting up my parameters here. So I can, for example, here I've got 2 hidden layers, each with 100 hidden nodes in them. I'm using an activation function, which is identity, but I can also use a logistic 10H or ReLU function. And I have a number of different algorithms here that update the weights of that network. So I'm going to use Levenberg here, LBFGS, and I can set how long I run it for. And if I want to repeat it, I can set replicable training as well. So that will randomize the data in the same way every time, so I can see if I get the same results every time. So the output of that I want to then feed into my test and score. And you can see it's run it, it was updating it, and now I can compare the neural network with the logistic regression, and it's not actually improving that much for this data. I can also try a support vector machine. So that's effectively trying to fit a line that separates out the two classes. And again, I can here in the support vector machine, I can use various functions, what are called kernels. I can use a linear function, a polynomial function, a radial basis function, or a sigmoid. So what that's trying to do is use different functions to separate out between, in this case, disease versus non-disease. It's trying to create what we call a decision boundary between the two different classes. So I'll use a radial basis function here for now. So I want to get the results out of that and put those into test and score. I can also use a random forest. So again, I want to try that. And so a random forest is like an extension of clustering. What it does is it randomizes the data and creates decision trees. And then by comparing the trees through different randomizations, then it ends up finding the best solution with that cross-validation, with those random subsets of data, using different random extractions of the data and different comparisons, and then building multiple trees and then merging them back together. And I think the last thing we'll try is probably a naive Bayes. So this is using a Bayesian probabilistic framework to come up with a model as well. Interestingly, you see, I can, if I don't know what I want to put this out to, I can just click and let the connection hang. So at the minute I didn't connect. I know I want to connect it to test and score, but I don't want to repeat that, so I can just drag it back up to this test and score. So now you see what I have is effectively a range of different methods for this data, and the file and select columns has run the same data, same selection of columns through each one. Now, what I might want to do is create multiples of this and try each variable individually through each one of these methods. And that would give us a more complicated workflow, but it would also help us evaluate the different inputs to the model. So once we have this, then we have a test and score and a comparison. Now, again, in evaluate, I can also do receiver operator analysis, so a ROC analysis. So this basically plots 1 minus the specificity against the sensitivity. And from this graph, the area we get under that curve tells us how good the classifier is. So I'll take the values out of there, and I can now create multiple ROC plots for my different methods. And again, similarly, I could create multiple ROC plots for each input that I have in the data. So you can see now I've got this workbench with a workflow. What I might also want to do is combine all of these methods together to stack, what we call stack them, to create an ensemble approach, which takes the values from each one and then uses a voting method to work out what an output is. So in model, I have this stacking function, and I can take output from each one of these, and I want learner to learner, okay. Yeah. So basically what this does is it takes the output from all of these and combines them. I can then put that into test and score and see if the combination of things works better than the individual thing. And you can see it's running through these now, updating the results as it goes. And so before I mentioned about how different algorithms might come up with different answers, this overcomes that problem. So what I want to see is how the stacker combines. And you can see here now that the best area under the curve is the stack. So what's happening is it's taking the output from all these methods using a voting method. So which one does it gives the best performance and which one gives the best result. And also it might be that some cases work well for one method and different cases work well for another method. And you can see that the stack has improved and gives us the best performance now. So that effectively tells us that we can get an ensemble that works better. We can also do things like from our data table here, we can feed in the principal components analysis. So this may break it, but no, you can only have one input. But you see now by effectively pre-treating with the principal components analysis, we can also see if that improves our data. Principal components analysis is a way of effectively cleaning up data, removing noise from it. So I'll quickly do those. I'm using all the stacker methods. Yeah, that's good. Okay. So now, it didn't really make much difference, but the let me just check that. Yeah. I know why. So I should really combine that with that. So I know why. I need the output from there as well. So I can run a pre-treatment for our data as well and pre-treat the data with the principal components before putting it through the workflow. It's taking a little bit longer. Let's see if it works. Looks like it's worked. Yeah, and now you see our stack has gone from 0.784 up to 0.797. So the PCA has removed some of the noise from our data. So that's really kind of an overview of some of the methods we can use in Orange for machine learning. I would say this is just the tip of the iceberg. There's not time to go through everything. But for example, there's methods here that we can use. So for example, image analysis, we can load in images and do work on those. What might be interesting from the group here is that there's a spectroscopy feature. So you can load in spectra and you can do, for example, hyperspectral profiling. We can do pre-processing of spectra, integration of spectra, partial least squared regression on spectra. And you can create a workflow effectively bringing files in to do that. So those can be used as pre-treatments before the machine learning as a method. Another area which might be interesting for you is the bioinformatics side. So we can bring in data from geo. So from the geo data sets, we can select a data set. It's taking a little while to load, but you can see here we can select from just about every data set in geo and load that in to our data analysis. So you almost can create a workflow which brings in a data set from geo, runs it through the machine learning, assesses performance, assesses parameters and works from that to find the best parameter set to use. We also have here a single cell analysis. So for example, we can download single cell databases. So this is a growth area. Single cell omics is massively increasing, but we can bring workflows to bear for that. So it's quite a simple method, but also quite powerful. As I say, it has limitations in that you can only sort of set so many parameters. You have only so many algorithms you can use, and so many things you can tweak and change. Whereas in things like Weka and more so in Python, you can do a lot more in terms of how you optimize and utilize different features. And for want of a better word, get under the hood and start playing with things. But it's a good starting point for learning. So that kind of brings me to an end of the demonstration. I would say, it's worthwhile having a look at this and having a go and just having a look at the data that you bring into the system. I'd be happy to answer any questions people have, if that's possible.
Sanjiva, are you there?
Hello?
Yes, sir. Tell me. Sorry.
Yeah, Ashwini, is there any questions? Can you coordinate the question?
Not at this time. I do not see any question, but there is one which is asked by Prajakta Biyani. Will you please elaborate about score and coverage coming after MS data mapping to database?
So, yeah, so if you're mapping to a database, you will get a closeness of fit score and a coverage score. So effectively, the closeness of fit is the error.
Because we've done a diamond.
And if the error is high, then the closeness of fit is not very strong. So you don't have a good match. But also coverage is a measure for the breadth of the protein or the proteome that you are getting with coverage. So it might be you have a very good score, but a very narrow coverage. It might be only for one protein. Whereas the ideal would be to have a good coverage of the protein and a high score. So you're getting a good fit and a large, you're covering most of the sequence of the protein. It's not really my primary area, but from working with SwissProt and combining sort of peptide libraries and getting coverage back from that's where I know. But others may be able to better answer that question than me. But that's my understanding of it.
Okay, there is one other question that I could see here, which is, what information does ROC curves gives you? I could not follow. That is something that they're asking.
Okay, so the point of the ROC curve is it tests a classifier's ability to correctly classify. So if you have, let's say you have a continuous value of predictions, you have predictions ranging from 0 to 1. And you can set any threshold in that to test your classifier. Now, normally you would set 0.5 as the threshold because that's halfway through the median, if you like. Now, what a ROC curve does is it varies that threshold and then tests the sensitivity and specificity of your classifier. So sensitivity, essentially picking up the true positives, and specificity picking up the true negatives. So what you want to know is how good is the classifier at a particular threshold of the decision. And so what you can then say is, well, if you have, let's say, an area under the curve of 0.8, which is normally considered an acceptable classifier, you have effectively accounted for 80% of the predictions and the individuals in the population. So you're predicting correctly for 80% of the individuals, despite the threshold of prediction that you're using. So the area under the ROC curve is effectively a classification performance measure. And the area that you ideally want an area of 0.8 or more, really.
All right, these were the questions that we had, sir. And there's one feedback which says it was a great presentation and highly informative. Thank you for such a wonderful presentation too.
My pleasure. Thank you for the feedback.
Really
Yeah,
wonderful
I think
presentation.
Dr. Sanjeev is back. So.
Okay.
It was really great presentation, Graham, and thanks for answering to the replies of the participants. I'm sure they will have more questions once they have gone through in some more detail. Given that we are slightly behind on time, so let us close this session. Let's thank Professor Graham for this wonderful lecture and interactive session. Next to that is now a quiz waiting for all the participants, and that is MCQ, so don't have to be afraid. Sushmita, are you there? Can we conduct that quiz session now?
Yes, sir. I can send the link.
So